Yes, good afternoon, everyone. Thank you very much for this kind introduction. Today I have a pleasure to present to you the newest development of Nisphero, Twin platform, a solution that we especially designed for culturing the spheroids, and the first immunosafety application that we developed together with our partners from Roche. But let's start with a question, why spheroids? There are a couple of reasons. Spheroids can be aggregated from the mixture of numerous cell types, including primary cells and stem cells. Therefore, they reflect well the physiological complexity of the tissues. Optimized protocol allowed to generate a reproducible, scalable, and reliable experimental model over time. And finally, with standardized tools, they allow for a variety of biochemical readouts and microscopy and integration in the established pipelines. Sounds like a really good compromise to me. Over the years, Nisphero has established 3D models for different organs, such as liver, pancreas, tumor, or brain, skin, and heart. What's more, we provided valuable models for human disease like steatosis, fibrosis, NASH, or diabetes. But at Nisphero, we are not only fascinated by biology, we are also a team of skilled engineers who design and develop easy-to-use, automation-friendly tools for high-throughput spheroid production and assessment, as Acura 96 well plate, imaging like 384 well plates, or more complex microfluidic flow systems. So due to those reasons, our partners from Roche approached us with their problem with assessment of biologics. Currently, the drug safety field relies on animal models, even though the biologics are species-specific, and we know about the differences between human and animal immune system. And even if a new drug passes the safety assessment, severe or even fatal adverse effects still occurs in the clinical trials. On top of that, animal studies are expensive, labor-intensive, not even mentioning the ethical considerations. So therefore, we aim to develop together a novel in vitro platform where we would co-culture tumor and liver spheroids together with immune cells so that we could recapitulate responses to the immunotherapeutics. Also, important factor for us was to integrate clinical relevant biomarkers, and our solution needed to be scalable, automatable, and cost-efficient. So we came with this idea, with the smart but simple idea of two interconnected wells, which allows co-culturing spheroids together, but analyzing them separately. Well, easier said than done, and this is actually the sketch from the very first meeting with our partners from Roche. But after 2.5 years of common efforts, today we can present you the Acura Twin plate and the immunosafety applications that we have developed. But let's see the design more in detail. So Acura Twin plate is based on the standardized 384 well plate format, so it can be easily integrated in your pipelines. There are 192 twin units in a single plate with cell-repellent surface that prevents cell adhesion and preserves spheroid morphology long-term. We have integrated sure exchange latch, which protects spheroids over multiple media exchanges and secures minimal residual volume. The well within twin unit are interconnected with microfluidic channel, which enables tissue-tissue interaction and migration of immune cells. The bottom of the plate is sealed with COP membrane that gives the best optical parameters for high-content imaging. And finally, the gravity-based flow system is generated by a tilter to support nutrient and cytokine exchange, but also make the parallelization of experiment easier. So in our setup, spheroids were composed in HCT116 colon cancer cells and cancer-associated fibroblasts, and they exhibit a compact structure modeling solid tumors. The liver spheroid contained not only hepatocytes, but also endothelial cells and Kupffer cells, and they maintain metabolic activity over time. Both spheroids were co-cultured together with PBMCs from healthy donors in the twin unit. As a model molecule, we have evaluated efficacy and safety of runimatomab, a HER2-bispecific antibody, which is currently phase 1 clinical trial. Bispecific T-cell engagers are engineered antibody combining a domain-recognizing CD3 antigen from T-cells and a cancer-associated antigen, in our case HER2, which is a receptor commonly expressed on tumor cells. The coupling of those two domains allows by the molecule to engage T-cells with cancer cells and facilitate lysis and polyclonal T-cell response by passing MHC signaling. An obvious clinical benefit of this technology is creating very potent therapeutic molecules, but with increased risk of on-target but off-tumor activity and systemic cytokine release syndrome leading to organ failure. And this feature made the BiTE molecule an ideal model for our system. So how an experiment in Akura Twin looks like. So firstly, we aggregate microtissues in separate plates, which gives us several advantages. We can use optimal aggregation protocols and media. We can pre-treat the microtissues, we can induce the disease, and most importantly, we can perform quality control. Then the spheroid can be transferred to the twin plate manually or in semi or fully automated way by a robot. At last, PBMCs are applied and compartment dosed, and experiments were run up to 7 days with monitoring tumor growth, measured as a fluorescence of labeled cells, and the liver conditions was evaluated by the release of liver aminotransferase ALT, a common clinical biomarker. So firstly, we assessed tumor killing efficacy with different PBMC number applied in the system, and we have observed cell-dependent tumor killing in both CD3, CD28 stimulated PBMCs here in orange and HER2-specific antibody here in green, but we did not observe any killing in naive PBMCs or control BiTE groups. From the exactly same wells, we evaluated liver toxicity measured as ALT release, and we observed cell number-dependent increase in ALT release for both CD3 and CD28 stimulated PBMCs and HER2-specific BiTE, but only at day 3 after the treatment, not anymore at day 7, despite the redosing of the compounds. And these observations suggest rather acute than chronic liver damage effect. In the next step, we evaluated the HER2-specific antibody efficacy in increasing concentrations, and we observed a dose-dependent tumor killing effect for the HER2 BiTE, here in green, but not for the control BiTE, here in orange. And again, we also analyzed supernatants from the same wells, and we observed dose-dependent liver toxicity for HER2-specific antibody, and again, at the day 3, but not anymore at the day 7. In order to assess the phenotype of the spheroids after the treatment, we performed the immunohistochemistry analysis, and as expected, tumor spheroids in CD3, CD28 stimulated PBMCs and HER2-specific antibody exhibited significant changes in tumor tissue composition, infiltration of CD3, CD8 T cells and granzyme-positive cells, but also staining for cleaved caspase-3 indicated induced cell death. And in no PBMC and naive PBMC groups, tumor spheroids remain intact. Interestingly, in CD3, CD28 stimulated PBMCs caused significant liver disruption, T-cell infiltration and cell death, and this effect was not observed in HER2-specific antibody, suggesting different than direct T-cell mediated killing hepatotoxic effect. So we hypothesized that the secretion of inflammatory cytokines by activated T-cells might be a possible cause of the liver damage in our model. So we evaluated the release of cytokines after the treatment with HER2-specific antibody, and we observed the release of T-cell dependent IL-2 and TNF-alpha already after 24 hours, and interferon gamma and IL-17A at later time points, 48, 72 hours. And secretion of cytokines was followed by the release of ALT with the peak of secretion after 72 hours. To summarize, AcuraTwin allows for combined investigation of tumor-killing efficacy of immunotherapeutics and assessment of liver safety. What is even more important, using our new platform helps to distinguish the mechanism of liver toxicity, either as a direct on-target immune cell-mediated toxicity or as an off-target result of cytokine release syndrome followed by the liver damage. And this effect would not be possible to observe in the monoculture. Therefore, I hope I persuaded you that AcuraTwin is a powerful tool for studying tissue-tissue interactions. And with today's launch of AcuraTwin platform and immune safety application, I would like to invite our industrial partners for collaboration. But we also need our academic partners to help us to unlock the full potential of AcuraTwin. So our academic partners can receive an evaluation kit to develop new important application for investigating tissue-tissue interaction and get our support to present their data at another scientific conference, maybe MPS 2024. And with this call, I would like to finish my presentation. Thank you very much for your attention. I will be glad to take any questions.
Thank you, Michal. And so now, if you have any questions, please feel free to ask.
Really interesting concept here. So bear in mind, this is really not my area of expertise, so the question may come across as a bit naive. But what would be the problem with actually putting the two spheroids in the same well?
It's not necessarily a problem of putting two spheroids together. However, they could merge simply, they could fuse. But separating them give us a couple of advantages. First of all, the separate microscopic analysis. This is the one of the advantages. And second one, due to the spatial separation, we can observe the active migration of the immune cell from one cell to the other cell, which could be also applied for observation in vitro of the abscopal effect for the localized treatment, for example, reprogramming of the cells or this kind of a treatments.
Okay, good points. I mean, I was wondering, so what if in the merging channel you were to include the, I suppose that you could modulate the possibility of immune cell migration by including some sort of barrier that would limit the size of what can go through in the connecting channel. So maybe also consideration for future development with and without that would allow you to discriminate cell migration.
Yeah.
Yeah. All right. Thank you.
I would have a question. So for this very complicated, very nice model, you used several human donors. With regard to this immuno aspects, how was the discussion whether you need maybe the same genotypes or donor specificity? What's about that?
Yeah, absolutely. This is a very valid question and very important topic to discuss here. So in general, over the course of development of the platform, we planned to use the matched donors from the hepatocyte donor and PBMCs. However, for the initial development, we use our standard model, which is 10 donor pooled microtissues. It came out over the time that we actually do not observe allogenic response. We don't see the release of ALT or ALT from the liver spheroid, and we also don't see the tumor toxic effect. Probably this is due to the relatively low amount of PBMCs that we have in the model, so we don't have the T cell that would specifically recognize the microtissue, and also we do not have the antigen presenting cells coming from the second donor, so the mixed lymphocyte reaction also cannot happen in the system. So I think for now this system works quite well, but of course we are considering, and we do have the means to match the donors and keep the system, for example, for only one donor.
Thank you.
Okay.
Great presentation. I have one simple question. Do you have any comparison between static and fluidic condition in aqua twin?
Yes, we do. Actually, we have observed, and we also have the supportive data from mathematical modeling, that without this tilting and enabling the liquid exchange, the diffusion is low enough so we don't see the significant exchange of the nutrients of cytokines. This is a plus and minus at the same time, because without tilting we can actually observe construction of the gradient from one well to the other well, and this could allow us to build the chemotaxis gradient, and we can see then the chemotaxis in the channel, which can be visualized by the microscope from the bottom. Thank you.
If I have time, I would have one last question for you. If I have 1
Sorry.
minute. It might be very naive, but I was wondering if there was an explanation, a reason for your test to end up at 7 days. Maybe it's a problem of PBMC, survival over time, or any other.
So in general, the longest time that we, first of all, cultured liver spheroids in the plate was 14 days to check the biocompatibility of the plate. The longest experiment with tumors were around 10 days, because the tumor is overgrowing and we start seeing the cell death and necrosis in the tumor. And this 7-day course gives us time to see the growth of the tumor, and also we are able to monitor the liver toxicity.
Okay, thank you.
Thank you.
Thank you again.